Good morning everyone. In today's module we are going to see plant vacuoles. Plant vacuoles we have divided into two parts, plant vacuole 1 and plant vacuoles 2. So today we are going to see plant vacuoles. Plant vacuoles are nothing but the intracellular compartments which arises as a terminal product of secretory pathways in the plant. Plant vacuoles are multifunctional and help in many functions, mainly in the temporary storage. Also it controls the turgor pressure and helps to maintain the cell shape. So we are going to study in detail about the plant vacuoles. Objective of these modules are introduction to plant vacuoles, second structure of plant vacuoles, biogenesis and development of plant vacuoles, and the fourth one, that is types of plant vacuoles. Introduction. Plant vacuoles. Plant vacuoles are multifunctional organelles which play important role in plant development. They are lytic compartments, function as a reservoir for ions and metabolites. The most distinctive feature of the plant vacuole is its size, which is usually occupies over 80 to 90% of the cell volume in well developed somatic cells. As per new definitions, vacuoles can be defined as intracellular compartments arising as terminal product of secretory pathway. They are ontogenetically and functionally linked with other compartment or components. Vacuolar contents are derived from intracellular biosynthetic pathways and coordinated endocytic pathways. The biosynthetic pathways include 1. Sorting of proteins destined from vacuole in early stage of secretory pathways, 2nd. Endocytosis from plasma membrane, 3rd. Autophagy pathways for vacuole formation, 4th. Direct cytoplasm to vacuole delivery. Plant vacuoles discovered by microscopy. Vacuoles are widely diverse in form, size, content and functional dynamics. A single cell may contain more than one vacuole. Usually a central vacuole occupies large volume and it's essential for many physiological functions. Electron micrographs of meristematic vascular cylinder cells and their vacuoles in tobacco root tips. Electron micrographs of meristematic vascular cylinder cell and their vacuoles in tobacco root tips at 2 to 6 days. A and E in 2nd D cells, the very darkly stained PSVS lack phytin globoids. These are protein storage vacuoles lack phytin globoids and appear fused into large multilobed vacuolar system. B and F in that 3rd D cells, the darkly stained vacuole are mostly round and their membranes are continuous with collapsed vacuole membrane, that is CVM. Domains are shown by arrow. C and G, the vacuoles in the 4D cells are of the protein storage and lytic vacuole or lytic vacuole hybrid type, and their membranes are continuous with very extensive collapsed membrane domains. The staining of the vacuolar content is variable and occasionally fragments of membrane like structures are also seen within the vacuoles. D and H. In 6D root, some cell possess a mixture of mature LVS with unstained lumina, whereas other contain pre-LV or LV or autophagic type of vacuoles with typically lightly stained flocculent contents and patches of darkly staining material. These vacuoles also show evidence of fusion of small vesicular structure with the large vacuoles. J. The boxes in G and H indicate the collapsed vacuole membrane domains shown in I and J. N is nucleus and scale is given. Vacuolar structures. Vacuole structures mainly consist of two parts: vacuolar membrane, that is tonoplast, and vacuolar cell sap. Now, here is the example of the cell's vacuolar structure. In that, Arabidopsis root cells in the elongation and meristematic zone. We are going to study the structure of vacuoles in Arabidopsis. Here, tobacco BY2 cell expressing GFT, ATV, VAM3, which is mainly localized to VM, that is vacuolar membrane in interphase and mitotic phase. D. Yellow arrows and arrowheads indicate transvacuolar strands, that is TVSs, and the tubular structure of the vacuolar membrane, that is TVM, respectively. Leaf epidermal cells in 1 week old Arabidopsis expressing GFP, ATV, AM3 VAM cells in E. Yellow arrow indicates TVSS structure, and F where hypocotyl cells in 1 week old Arabidopsis expressing GFP, ATV, VAM3, AT-VAM3 spherical VM structure are bulbs. In this figure, we have seen example of vacuolar structure A and B. Arabidopsis root in the elongation where A and meristematic B zone. Vacuolar lumen and cell wall are stained with CF green and propydian iodide, that is red, respectively C and D. Tobacco BY2 cells expressing GFP, AT, VAM3, which is mainly localized to the VM in interphase, which is C, and mitotic phase D. Yellow arrows and arrowheads indicate transvascular strands, that is TVS, and the tubular structure of the vacuolar membrane TVM, respectively E. The leaf epidermal cells in 1 week old Arabidopsis expressing GFP, ATV, VAM3, yellow arrows indicate TVSS. Hypocotyl cells in 1 week old Arabidopsis expressing GFP, AT-VAM3. The spherical VM structures are bulbs. The membrane surrounding the plant cell vacuole is called the tonoplast or vacuolar membrane, VM. The membrane of protein storage vacuoles contains a seed specific aquaporin called as alpha TIP, tonoplast intrinsic protein. On other hand, other storage vacuolar membrane shows presence of gamma TIP. The membrane of other specialized vacuole shows presence of specific transporters like ATP binding cassette transporter, that is ABC transporter. Immunogold localization of alpha TIP and gamma TIP in the tonoplast. immunoglobulin localization of alpha TIP and gamma TIP in the tonoplasm membranes of inner cortex cells in 3, 4 and 6D seedlings. A and B shows PSVS of 3D plant whose tonoplasm membranes are strongly labeled, shown by arrow, with anti-alpha TIP antibodies in A, but not anti-gamma TIP antibodies in B. Some labeling of vacuolar contents is also seen in A. C and D shows collapsed PSVs, that is protein storage vacuoles membranes of 4D roots labeled strongly, shown by arrows, with anti-alpha TIP antibodies, shown by C, and weakly, which is shown by arrowheads, with anti-gamma TIP antibodies in D. E and F in inner cortex cells of 6D seedlings, virtually all the alpha TIP labeling has disappeared from the newly formed LV membrane. In contrast, the gamma TIP labeling of the LV membrane is significantly increased compared with the 4D samples. In F, here CVM is nothing but collapsed vacuolar membrane. Biogenesis and development of vacuoles. What is the origin of vacuole? Difficult to determine because many vesicular compartments in the cell are similar to precursor of vacuoles or provacuoles. This is the only organelle that is produced de novo, that is not through inheritance. Most likely the trans-Golgi network TGN and smooth endoplasmic reticulum SER produce provacuoles that develop into mature vacuole. Techniques used to study the biogenesis are TEM and molecular marker labeling. In the meristematic cell, only very small vacuoles or provacuoles exist. As the cell expands, small vacuoles fuse to form larger vacuoles and eventually form the central vacuole. As the cell differentiates into different types, the vacuole develop into various functional groups as well depending on the cell type. Model for lytic vacuole biogenesis in Arabidopsis. Origin of vacuole is difficult to determine because many vesicular compartments in the cell are similar to precursors of vacuole called as provacuoles. Most likely the trans-Golgi network, that is TGN, and smooth endoplasmic reticular, that is SER, produce provacuole that develop into mature vacuole. Techniques used to study the biogenesis are mainly TEM and molecular marker labeling. In the meristematic cell, only very small vacuoles or provacuoles exist. As the cell expands, small vacuoles fuse to form larger vacuoles and eventually form the central vacuole. As the cell differentiates into different types, the vacuole develops into various functional groups as well, depending on the cell types. Precursors of vacuoles, the provacuoles, arise from the endoplasmic reticulum, that is ER, via maturation. The vacuolar proton pump VHA-A3 and AVP1 and perhaps the other tonoplast proteins are incorporated to the nascent provacuole directly from the endoplasmic reticulum membrane through an uncharacterized mechanism shown by a lower right corner. Impairment of Golgi and post-Golgi trafficking, which is shown by cross arrow, leads to aberrant multilayered provacuoles. These observations could be explained if impairment of Golgi and post-Golgi trafficking would interfere with the process of provacuoles release from the endoplasmic reticulum or with the fusion between provacuoles and vacuoles. Biogenesis of protein storage vacuole. In this diagram, it illustrates the ontogeny of protein bodies, that is PBS, and protein storage vacuoles, that is PSVS. PBS from through the aggregation of storage proteins within the endoplasmic reticulum, or PSVS. After formation of PBS can either remain attached to the ER or bud off and form separate organelles, that is PSVS, that is protein storage vacuoles. PBS can accumulate in the cytoplasm or become sequestered into PSVS by autophagy. PSVS are formed as the consequences of endoplasmic reticulum synthesized storage proteins progressing through the endomembrane secretory system to specialized vacuoles, that is PSVS, for accumulation, and this is given by Herman and Larkins in 1999. The development of mature vacuoles is probably the result of fusion events between provacuoles, autophagosomes and vacuolar transport carriers. In this figure, first A, immunogold labeling of ultra thin section of high pressure frozen freeze substituted and HM20 embedded Arabidopsis pad2 root tip cells shows a provacuole, which is shown by the dashed red line, carrying the VP phase VP1 adjacent to the provacuoles, a ring like structure which is not labeled by the antibody, that is alpha AVP1, could be an autophagosomes. This picture shows that at least two distinct populations of circular or semicircular double bilayered structure exist in Arabidopsis root tip cells. And the B shows the development of mature vacuoles is probably the result of fusion events between provacuoles, autophagosomes and vacuolar transport carriers. Types of plant vacuoles. A single cell may contain more than one vacuole. Usually, a central vacuole occupies large volume and is essential for many physiological functions. On the basis of content and functions of vacuoles, vacuoles are classified as follows: central vacuole, second protein storage vacuole, third specialized vacuole, and fourth lytic vacuoles. Central vacuoles. The central vacuole is the largest cellular organelle found in plant cells. It is surrounded by a membrane and functions to hold materials and waste. It also functions to maintain the proper pressure within the plant cell to provide structure and support for the growing plant. Protein storage vacuole. The bigger vacuoles are protein storage, which is shown by V1, and the small ones are lytic vacuoles, that is V2, in single type of cells. Protein bodies are protein storage vacuoles found in seeds and which has maintain their pH, which is shown in the figure. Specialized vacuoles. Specialized vacuolar compartments present in variety of tissues. In some tissues like leaf, epidermal cells shows presence of defense or signal compounds in vacuoles. Pigments like anthocyanin are stored in vacuoles of epidermal cells from flowers, leaves and stem. These specialized vacuoles helps in storage of secondary metabolites of the plant cells. Lytic vacuoles. A vacuole that is maintained at an acidic pH and which contain degradative enzyme including a wide variety of acid hydrolases. Those vacuoles are called lytic vacuoles. A simplified map of the secretory pathway of a higher plants shows the cargo synthesized and processed in the endoplasmic reticulum ER is transferred directly to the closely opposed and mobile Golgi apparatus G. However, direct transfer of some storage protein to the vacuole may occur by route 1. Proteins destined for the protein storage vacuole may leave the Golgi at the cis and trans compartments, which is shown by route 2, while the exocytotic cargo predominantly lives at a trans phase, that is by route 3. The product destined for the lytic vacuole is sorted at the trans Golgi and routed in clathrin and receptor dependent fashion through a pre-vacuolar compartment, that is PVC, which is shown by route 4. Endocytosed materials and recycling plasma membrane proteins are internalized into an endosome like compartment, which is shown by route 5, which might transfer material to fuse with mature into the PVC. Route 6, which is shown by route 6, the exact role of vesicles in the transfer between putative endosomes. PVCs and a lytic vacuole is unclear. This is the role of lytic vacuoles. To summarize these modules, plant vacuoles are very important part of the plant cell system. They plays important role in to maintain cell shape, turgor pressure of a cell, again also help in membrane transport and work as a temporary storage. Plant vacuoles are of 4 types as we have studied. Central vacuoles, which is a large vacuole, protein storage vacuoles, which helps for the storage of proteins, specialized vacuoles, mainly the function of this vacuole is to store pigments, secondary metabolites, etc. And the fourth one, that is lytic vacuoles.